making sure. Perfect. Okay. So, a few important points for safety here in the lab. Also to prepare yourselves whenever you're in a lab down in the road in your future. A few important reminders. You wash your hands down there at that sink. This sink is for portable use when you're cleaning up equipment. If equipment is sink where you clean up your hands. And so here, you have eye wash station here. If for whatever reason something gets into your eyes, always when they goggles. If for whatever reason, Ameen and you can have the eye here. Switch it or as you turn it, and then you come into the lab, you have your lab coat nicely mixed in with the button clipped on. And for whatever reason, let's say if you're using buttons and turns in the lab, of course we don't want to get hurt, but if for whatever reason something happens, you can actually turn this around and turn on the shower. So if this is in the shower, there's a glass blue bin right here, where you won't be using this lab as much unless you bring glass. So just bear that in mind. The red bin here is just for garbage, just like the one shown here. We'll be opening the middle row for any extra items. The microscopes are already present on your benches, but for whatever reason, if you need additional equipment, if let's say your microscope is not functional, we'll make use of opening that up. We have depression slides and many equipment that we'll be using today already here in the middle bench. So bear that in mind when you come in either as groups or as pairs or individually, that you make sure that you make use of the equipment found in the middle row, but you also return it back in its former state. We don't want to have any depression slides or any used or reused items to be thrown in the garbage nor in the glass. So just bear that in mind, everyone. And that's about it, apart from, keeping in mind not to eat or drink anything in the lab. If you need to drink or eat, just let me know and you can do so outside. If you need to use the cell phone, also let me know. And before we begin today's lab, I would kindly request, you'll see, I believe, a red or transparent bottle for 20% found on most areas across benches here. And so what you're going to do is begin the lab by just bringing your bench, so that way you maintain a static technique. Before I even move further, any questions? Any questions? No questions? I didn't say this early on. I'm trying to be fast-paced, but something that's very important for me is the concept of being able to integrate your knowledge in different ways. So, many of us are maybe in our third, second, fourth, or fifth year here on campus. And so I think it's very important that when you come into the lab, you learn how you can understand and apply concepts more readily once you leave it. And so it could be, the stains that we're learning to use today, maybe it'll be beneficial not just for your resume, but when you learn about how these different techniques can be applied, let's say, to medicine in a clinical setting or in pharmacology and other fields as well. So just bear that in mind. I also appreciate, and hopefully all of you have seen my, at least some of the skill sets I've referred to in the past of punctuality, coming in on time, organization, keeping things neat in your lab notebooks. And with that in mind, if you haven't shown me a microscope in your notebook, like set up with the annotations, please do so next week. I want to make sure everyone comes in, not just with the best of standards, but being able to best succeed in this course. I want all of you to excel and get the best mark that you can. So just do your best to put that effort in, and inshallah you'll see the fruits of your endeavors soon. That looks like it from there. When you look at your lab manual, you'll see an outline whenever you come in, and you'll see, these are the exercises I'll be doing today. And many of them we'll try to complete as much as possible, inshallah. And I think data analysis is shown here. Assignment, you have no assignment for this week, but in 3 weeks' time, you'll be having your unknown assignment. So think about how you prep for staining today. There's 2 to 3 main concepts that we'll be focusing on, and that's exercise 1, the light microscope, exercise 2, which actually I believe you'll be doing yourselves, And then exercise 3, actually preparing yourselves with a smear and staining for success for the next few weeks. Any last questions before I move further? No. Okay. And don't feel shy because I don't mind answering more questions. In terms of the microscope, I said this earlier, but this is the type of microscope that you want to draw, and is pretty much like the microscope that you have on your desktop. So whatever you see on the microscope right now, have a look at it, try to identify the different pieces, be it the nose piece, the stage, the condenser, and so on. And especially when you're moving between objective lenses, right, from like 10x to 40 to 100, you need to bear in mind that you're able to, prior to this, focus your specimen in place. And so that's where you're using the coarse adjustment knob to move more quickly up and down your stage with your specimen on top. And then you have the fine adjustment knob when you're trying to make sharper tuned focuses or fine adjustments to your focus. There is a question, I believe, in the next slide. And here it is. So immersion oil. Who knows what it is and why we use it for light microscopy?
Yeah. Like what you put on, slide, which increases the refraction, some more light.
Okay. You also had hands. Okay. So, okay. So, any other thoughts? Any pointers, ideas? Pretty much this is a good concept of integration. Both ideas are right, and in fact, both couple each other quite well. The idea is that immersion oil, not shown on the left-hand side, but shown on the right, is what's able to essentially provide greater refraction of your light rays, which means essentially that you're maximizing or optimizing the amount of light that gets captured and passes through your specimen, shown in the red rectangle. And that way, as you take images, or sorry, you're not taking images here per se, but as you visualize your specimen with light microscope, you'll be able to maximize your resolution at 100x magnification. So just bear that in mind. Many questions that you're asked throughout the duration of our practical will be noteworthy for your better understanding of the material and your preparation for the lab exam. So here is pretty much a demo of how you would make use of oil immersion. It's about 5 minutes long, but we'll be going hopefully to actually showing more practical ways of you being able to, insert the oil immersion or immersion oil. Very simple though. The idea is that when you're between objective lens, and if you need help, I'll show you, between 40 to 100x, once you've focused your specimen at the given location on the stage, you pretty much add in that drop, just one drop of oil immersion, and then you slowly tilt your objective lens so that way it reaches back to that 100x objective lens to ensure refraction is optimized. You add in, yep. So that's pretty much what the question situates itself as. If you lose focus, then what you need to do is just tilt. Ideally, you go back and repeat the steps. But what you can do is go to 10x rather than repeating the entire process again. You essentially, I'll show you with a demo microscope. I'll show you on this one. Let's say whatever reason. This is where you normally add in the oil immersion, and then you are already focused, right? So you've already adjusted the location of the stage. But let's say you added in, not sure to your but the oil immersion which you added in here, and you focus, now it should only be steps such that you don't have to adjust once again, especially the course adjustment knob. At the very minimum, find the if for that reason though, let's say like something happens, the stage is on down, then what you're going to do is go back to 10x, repeat the process, and then slowly go back to 2x. You can more professionally go and 40 and 10, and then press so fast after you've adjusted the stage. For your slide, light microscope If you need help with like, actually loading the microscope, here's my suggestion. You load it from the top or by the neck, right? So what I'm showing with my dominant hand, and then the left hand or your left dominant hand with the at the bottom. So just make sure that you try to hold it carefully. And bear in mind that if you need your microscope is not essentially aligned with my focusing, then you should manage that. So let's say I'm focusing right on my objective and I'm not 40 x right now, I'm going to get a high resolution. I'm going to add in one drop of oil version, then I'm going to slowly move from 40 to 100x. And by that time, the stage should have already been adjusted, so that way you could have the specimen like high resolution. Now, if for whatever reason the stage drops or you have like something happens such that you're not able to focus, and pretty much answering that yellow bubble question, you go to 10x, and then you just go through the process of adjusting the stage, and then you eventually return back to 100x. Normally, you won't need to add an additional drop of oil, but if you see it dried up, then that's where you do so. When you do clean up, though, after the end of today's class, make sure that you clean the bottom lens with 70% ethanol, so that way you don't leave a mark for the upcoming practicals. Any questions though on that note? No questions? Okay, so let's do. Let me just see timing. Okay, so I think this was the recommendation from Dr. Brunt. Let's do smearing, or actually before we go forward, aseptic technique. I mentioned it earlier, but I'm going to show you a bit of a demo. First off, before you begin the lab with the Bunsen burner, you need the gas to be on. There's the gas valve right here. Only I'll be putting it on, so you're aware. Over here. Now it's on, essentially a line parallel or pipe. Now that's on, meaning that be aware that once you turn on or open your own individual blue valves for gas, that you can actually start a fire. So bear in mind that you keep it clean safe and that you don't run the risk of endangering yourself and others. I may need. I want to make sure you're all safe, so I'll just give you so always make sure you do 2 steps first. Right before you even begin, you spread or spray ethanol, 70% ethanol on your bench, and then thereafter, such that it will be completely aseptic technique, meaning essentially that you're reducing the risk of septal bacteria that will essentially contaminate your bench, specimens, and itself. So the idea is that you're going to spray this, right? What I typically use like an S curve or like imagine you're drawing in person. So something like that. You have to put the chair, but the point is that you clean up your bench pretty well. Then, while keeping in mind that you're going to start fire and start your Bunsen burner, bear in mind that the ethanol is not too close because this is flammable. So you really don't want to make any fire come out. As you turn on the gas valve, you just turn it slightly open. So for many of you, it will be either counterclockwise or clockwise. You'll hear a little bit of a sound feed out, and then start to fire. So it's important that bear in mind 2 things in this period. Hopefully you can all see. Can you see from this side? Yeah. I'm going to start first now, and then just lower it again. And I'm going to do the same for that time. The point is, you have a fire now, right? It's okay how it's starting, but it's important that you always keep an eye on it. You don't want to have like fires going in and out. It's a very similar process. And that's pretty. That's what we want. And normally you'll have a paper towel. But you need to use paper towel. Right? And essentially you do the same process of making sure that you're aware that your ethanol is not even close to, not just the pipes, but other flammable materials that could, ignite that fire even more. So, that should be tilted slightly. So now, I didn't even hear much sound. So, what I think, at least for this bench, this one is the gas. Find that the gas isn't turning on too well, that could be a sign that you may have to switch. Like the blue ones on the side should be good. And if, like in my case, look at it, looking for just a bit. At max, do it to half of the tilt. Don't do it the full way because it can actually run the risk of burning you. Just bear that in mind. So as you can see, it's halfway and it's already quite violent. Go down. So we have to be very careful. As you can hear, it sounds like a rocket, right? So just be careful if you don't burn yourself. And the innermost cone, is yours still on, by the way?
Yeah.
Yeah. So the innermost blue cone is the most flammable location. So bear in mind that when you're sterilizing your inoculating, you're going to be actually, I'll show you this slide. You can actually place it in for a few seconds until it turns flaming red. Now you can see it's quite red at that tip, that edge. You want to make sure that, yeah, right there. Just like that, it's pretty red at the very center tone. So, or that circle. So just bear that in mind when you begin any lab and you're using bacteria, then you're going to be flaming two things. The soup itself, the bacteria that you put in, and the inoculating loop before and after, both times. Right? So bear that in mind. If you're doing it before, also bear in mind that it's not too hot for your loop, so that way you don't pull that bacteria that you have, and then you can't use smearing. Any questions from your side? I'm going to repeat the process. I'm going to also shut it off so you guys can do this process again, and also so that the fire or the flame doesn't spread too vigorously. The point is what I showed them is that when you're starting off, any experiment, bear in mind two things before and after each time. If you're using any tube, liquid, solid box, that you're going to be cleaning the lid itself and your inoculating loop. When you're cleaning the inoculating loop, it should turn like how it is now, red or golden, essentially at the loop itself. But bear in mind that it's very hot, right? So you need to be careful that you don't actually go straight into your tube, otherwise the bacteria that you want to clean and actually visualize. So when you're done that process, that's where you repeat the process of at least at a minimum cleaning the tube again, and that way you don't get contamination. That's pretty much a septic technique for you. Sterilizing your bench, keeping your bacterium as sterile as possible, and also making sure that any transfer of materials in and from your tubes is always sterile. Any questions? Question? No problem, no problem. And don't feel shy. I love questions. So that's pretty much this. Simple staining is what we're going to do now. And so I'm just trying to think, because once upon a time we do that whole thing. So I think I may start with, or it's better maybe be more equitable, more fair for everyone, all the options right now. You'll have 3 slides, so bear that in mind. 3 slides for 2 bacterium, and this is for the simple state of what I believe is not just states that you for the simple state found here, saffron, red, crystal violet, and pretty much blue or violet, and we have methylene blue as another type of state. You really need to be using for simple state 2 out of the 3. So just bear that in mind. And then when it comes to negative stating, which we can use if you have time today, we'll be using nitrogen. Pretty much you'll find the state that you need on your bench, so don't worry about it too much. But in the meantime, let's prepare ourselves all in advance with starting off smearing our bacteria. I showed you pretty much what you need to do to actually prepare yourself for smearing. The point is now what you really need to do is have a labeled slide, which not from your function, but here. Let's see. Not only slide. No slide, things like this. Off that segment. I just don't know if we have enough for everything. So at max, then use this 2 slides per person or per here for the exercise. I believe this is done individually, so 2 per person should be enough. 2 per person. Because I don't want you to 4. This should be enough. So start off with and actually make use of just one slide at a time, so you can keep one as a backup. But the point is you'll be using 2. You'll be making 2 different smears using the liquid and or solid broth. Solid is the one that's out of plant, looks like it's almost frozen, and then the liquid is the one that's liquid and you move it about and see viscosity. What's important is that you're bearing in mind from the lab manual how many drops of water you're adding and how much of at least solid agar you're going to be adding in. It's very, very small. Is your thing still on?
Yeah.
Yeah, okay. So I'm just going to show it this side and then I'll show you guys as well. It's very simple, but you want to bear in mind that you're probably trying to notice the amount of bacteria you add, so that way you don't run the risk of essentially over smearing. Can everyone come from for timing, actually, if you guys all so I'm going to repeat the process. I think this is the same. Only going to be right, especially I don't see. You can come around. Maybe instead of using the shirt, one stand up and have way more people can but it's a very important concept because sometimes people can, lose time not making the right decision. So imagine this is your agar, right? And you have let's say I have on the slide, I will label on the left hand side the 2 sets of bacterium I'm using and the simple state to make it myself. So now imagine, let's say we begin for example. So imagine I'm ready to start. And now I'm going to be labeling my inoculation first because it takes a bit more time. So take time to have it turn red. And that'll take a bit of time to pull it down. So here's my left. Sorry. So imagine this is lit, or sorry, the tip of my other tube that I'm going to be inoculating through. So now I'm playing the top to run the risk, to reduce the risk of any bacteria going through. And hopefully by now it's feel like it hasn't touched the edge of glass, and it's just like it's smoke. I don't see smoke, so hopefully it's good to go. Now I'm going to go inside. You have two options. I would advise you going to, I would say, 1/6 into the tube itself, the solid agar, because you only need a small amount. And how small? That is, and that's very small. You probably can't even see it, but that's how small it really needs to be. Very, very small. So and it's really, it looks like you're just blemishing it at the very tip. So don't expect too much to take out. But what you're going to have, what you hopefully have done before that is on your cover slip, sorry, on the slide, you would have added 2 drops of water. So that way, what you're going to do is just essentially rub it through your specimen and not be able to dilute accordingly. Once you're done, imagine then you've actually completed that solid smearing of your bacterium, and then once again, wait until that it's cool, or wait until it's heated, and then you can repeat the process for any upcoming. Any questions on that? No. Actually, I know when I learned about this stuff too, because the point is actually with gloves, they are more likely to get flammable. So run the risk because especially if you have gloves that are too close, you could actually get yourself occupied. Bear that in mind. And also to look at some people also very mind. But now we're done. You repeat the process for your other slides and for your other specimens. You have a list of 4 solid bacterium found here. Bear in mind that you use the right one, right? You want to take the best uses with the most appropriate morphologies for each bacterium. And then after that, talk about some other things about, measuring your full use, essentially your oscillator units for calibration, exercise 2. So we're moving ahead to exercise 3, sustaining, and then we'll move back to exercise 2. In about a month. Any questions? Yes.
How many do exercise can you be able to measure for like bacteria?
And that's what we're doing that. We're going to combine. Yeah, I know some thought I didn't do this class or like didn't do it before, but essentially the suggestion from Dr. Brent, and it makes sense to save time for each and every one of you, and also to readily apply and integrate your knowledge as we will show pretty short. Any questions?
Final question. For the smear, you separate between the 2 lines per question.
Yes.
So
Yes. I'm just going to see if we count the slides again. If not, we could also do. Actually, as a suggestion to make it easier, let's start off with 1 slide per person. That'll make it even easier for you just to really compare and start to know how to stain and smear it. And then once you've done that, if you feel like you have more time for everyone else, then you can do a second slide. And likely we'll do this for, I think then we'll have more time even to go to calibration, then to the hanging drop, and then if needed, negative staining. Any last questions before we move further? Any questions? No? Yes. Yes, you can. Now here's the thing. Dr. Brent does not want you guys to use lab manual. Generally speaking, like in the lab, it is best that you actually make your lab notebook as descriptive and as visual as possible. So that way it's pretty much your own lab manual. So think of it like that. Try to be as descriptive and as informative for yourself as possible. So that way Dr. Brent comes in, she's not going to tag you and say like, oh, why didn't you, make your lab notebook more detailed? So just bear that in mind. For this week's lab, I don't mind you using it. Just make sure though in upcoming labs, I'll also be keeping in track your notebook and its quality. So bear that in mind. But for this week, I haven't penalized anyone, so just bear that in mind for the future. Any other questions?
Last question.
Yes.
That jar or whatever that is over there,
Yes.
is that what contains the 95% alcohol?
That's correct. So we're not going to really need it because the slides are already clean. They're already essentially pretty much new. If though you want to clean up one of your slides in the future, at least this is where your 95% ethanol will be. So just bear that in mind. Here they're only shown, I believe, in the middle bench, but when we use them for upcoming practicals or labs, we'll be using this more often, and you may find them in different groups. How does that sound? Sounds fine. Sounds fine. So you're ready to go. All the best, everyone. No problem. Yes, of course. So, sorry, exercise 3. Exercise 2. For exercise 2, you'll be working as blocks, so sets of 4, I believe. And so I'll show you what we need to do during that time. But don't worry, continue to focus where you are right now.
We said make 2 symbols on one slide, so can we use different bacteria?
Yeah. Different bacteria, by the way. Ideally, you can have your solid bacteria diluted on water in your first smear, and then the second smear with liquid, with liquid culture, and that may make it easier for you. Yeah.
Just the one or two?
Yeah. So the left-hand side, the clear side of the microscope side is where you actually write the game wall. So just bear that in mind in the case. Just you can use the left-hand side to break down, let's say, smear 1 as the given bacteria and smear 2 as the next bacteria. Like smear 1 or S1, your solid HR bacteria name, and then the second smear for the second bacteria.
So the two will be on the.
Yeah. And also don't forget your name as well, because afterwards, when we're going to be drying or warming up the stain or the smear, sorry, we're going to be using the middle bench. There is slide warmer here. So just bear in mind that you use the slide warmers when you're going to reach the next few steps following smearing. So once you're done with your smear, you're going to dry it up right here. So just bear that in mind, everyone.
Yeah.
Also bear in mind, no flames should be left unattended. I could likely smell gas, but just bear in mind that if I do, I'm gonna have to shut off a given we're weeks back.
Yeah.
So for drops, you can add one drop for your second smear and then 2 drops for your first smear. Yeah. Yes, absolutely. Don't forget though, hopefully you've ethanoled your banks, clean up your banks, right? Because you want to make sure that when you're submitting any upcoming assignments, you want to make sure that your bacterium are contaminant free. Sounds ironic, but the point is that you want to limit that risk of other bacterium getting into your first drop, first smear, 2 drops, second smear, 1. Don't forget also that you've flamed your tube. Don't forget that everyone. Everyone okay? So on the basis of two,
First one, solid. Very cool.
Yeah. Yeah. Sure, yeah, mix it in like water.
So everyone, I just want you to pay attention. For the liquid broth, for the liquid culture, you want to add at least, I would say a max 1 drop. I know sometimes it's difficult, but if you can, that's okay. Don't forget though to shake your liquid agar, sorry, liquid culture. Sometimes the bacteria gets suspended at the very bottom. If you haven't done this and you're already past the step, it's okay. Don't worry about it. But I would just say if you haven't tested up yet, continue
Yeah, that's all. We're just doing two different batteries, so we saw it and liquid. Yeah, this one. It's not stuck to the side. Can I see the others? The liquid. We have solids too. Let me see. Oh, they're all solid, right? Yeah, you need liquid. I'll get this for you, Metro. That's all. Okay. Yes. Need the water. That's the water. Sorry. Oh, we got there.
Yes.
Yeah.
Yeah. Yeah.
Here, let me bring it.
So, I'm good. Everyone, can you pay attention, please? I've been asked this point before, and I just want to make sure, and I apologize if I'm clear, but after you've added in your smears for your bacteria, you're going to be warming them up here in the slide warmer. This is how you're going to let them evaporate the water, but also to coagulate your bacteria on the slide, the heat fix potentially. Now, the problem is how you can visualize. You need to stage. So that's where you're using the slide tray to actually stage your and blotch only like 1 or 2 drops at max with those colorful stains that I mentioned earlier. Crystal violet, or methylene blue, or saffronish. And they have different time points of how long you need to keep them. And then essentially you'll be able to wash them off after about 30 seconds, at max 50 seconds. So just bear that in mind. There are details like this in the lab manual. If you need me to show it as well, just let me know, or you can ask one of your peers. But just bear in mind that you want to have this done before you go into the microscope. Well, it's on.
So you just bring our slides over.
Yes. Sorry, yes. Thank you. Are we letting our computers dry up? It should be about 3 to 5 minutes.
That's supposed to be good.
Yeah. I use it too much.
Use it.
Yeah, they use it synthetically. Sorry.
Yeah, it doesn't have to be a lot of color, but mine's a small drop. It's okay. That's all right.
That's it.
I don't know if I did this right.
And that was your solid or liquid?
The big one's my solid, the small one's my liquid.
That's good, that's good. So from each of you, can I just confirm, how many of you are not yet at the stage of drawing your slides? Please raise your hands. Okay, okay, that's okay. Now in the meantime, we're going to be waiting for those that have done so about maybe two more minutes for many of you to let your smears dry out. This is a process known as deep fixing, because the idea is that you have different options, either to flame and dry for a quick fix, or to let it slowly dry off. So that way you evaporate any extra fluid on your bacterium or bacterial smear, and such that it fixes itself onto your slide for visualization and for staining. So I hope, hopefully soon, inshallah, you guys will be able to complete the task. Don't see as pressure. Sooner we're done, better, and then we'll move forward more staining, and then
But I think, yeah. I don't think that's all.
yeah.
Okay, I just finished it. Try to recognize it. And I can just please.
Yeah. Some of you have slides that look nearly done for drawing. Highly recommend picking them up now. And then you can proceed to using the slide trees. When you're using the slide trees, that's where you're going to be actually placing your stains for about 30 seconds to 50 at max for different stains that I mentioned earlier. You have saffronin, methylene blue, you have crystal violet. So hopefully in due time, you'll be able to stain with just a few drops, by the way. You don't have to add more than 2. So just bear that in mind, and then let it suspend on that tray that you have. Oh, yes. It looks like it's this one maybe, yeah, this one may take time. Actually, so I would say place it bit. Which one is that? Actually, go ahead. It should be. Yeah. Yeah. No. So you're adding not too much. For staining, everyone please pay
Mine's not
attention.
looking too good.
You're only adding 1 to 2 drops. because these are bacteria, right? Microorganisms. One of the lab techniques will say like how to like a few drops like for so issue is I don't know how security
This is mine, yeah. What's my other?
Okay.
Okay. Yeah.
So that's okay. That's two. And then you can just wait 30 seconds. I recommend taking another stage though. Yeah. Now, in the next 20 seconds, you're going to rinse them off with water.
That's water, okay. Yeah.
And then rinse it up. So it's just like.
Okay. 1 second. Thank you.
So, yes. Be careful, everyone, that you don't stain yourselves. And just bear in mind that after 20 seconds, you tilt your smear or your slide. So that way you can just drain off the excess water, or sorry, stain, the dye on your smear. So if anyone needs help on that, just let me know and I can help coordinate. Should be. You might hear it before and there could be
Yeah.
Sorry, how can you?
We still do it.
Now, yeah, now it's hard, but if you get the zero, it's just releasing it now.
Actually, I don't know how many people were still on the for trying. Everyone's trying. Keep this, it actually also goes straight. And like, how long you can try it on the week for a week. Yeah.
Yeah, you see one.
Water? It's okay, sorry. Thank you. Yeah. Yeah. It's not.
You don't need cover slips for now, by the way, everyone, especially after you're staining, you're gonna be able to readily see it through your microscope. So don't worry about that.
Yeah. Mine is too, don't worry. It's okay, mine is. Yeah, just
I forgot to say this, but, or actually, it is implied from the last manual. Bear in mind that when you're blotting, you don't have to like touch the surface of your stain because there is stain that you want to keep intact on your slide. So just bear that in mind. Normally, when you're washing off the excess stain, then what you're doing is taking it from the edge. You're taking your slide by the edge, you're tilting it, and you're adding in water. So that way it just drains naturally. And you don't have to do it vigorously such that all the stain comes out, but you want to keep that which can remain on your smear as ideally as possible. Now let's say you have stain on your hand. Before even you wash your hands, there's something important. Very, very important. Not ethanol. Okay, but there's something important to bear in mind. If you get stains on your hand, you have this with kind of like a lotion to erase or remove as much stain on you. So bear that in mind on your dry hands. You don't have to wash your hands. Just take it out, almost like Vaseline, and then you just rub your hands. Ideally, you can do it now or at the very end of today's class. And then you just keep rubbing, and then after you wash it with water, you can turn that in more, or that is. You can do it also at the end of today's class, so don't worry about it. Just if you have more stains.
Thank you so much for.
Yeah, yeah, I would say so. Yeah, it is dry wood. I think if you call the water, I think at that edge, the anytime for you, closer to you, you may find there to be a very double towards you. So don't worry, but just kind of, I would say, um, continue with the anytime.
Thank you.
No time, no time.
What is 5 and 16?
Perfect. Wait, can you please, sorry, number five? Number six? I guess number five?
Medina. Yeah.
Yes, but for exercise science, I find it helpful. You know that we have a brain question?
Right.
Oh, that's you?
Yeah.
Okay, so perfect. That's okay then. Thanks for that.
Like I was saying.
Yes, that's one. Perfect. Hi.
It may be, microscope, coloring. There's such a thing as dinosaurs, or, yeah, dinosaurs, like in terms of like the range. Dinosaurs,
Diet stabilis?
like,
Are you saying for the flagella? Or,
yeah, I mean, when you put up your range.
from the flagella, right? Oh, sorry.
No, not the flagella, I mean, from my screen, I see like,
Oh, so they can have an arrangement where they're in pairs. That's correct. But you wouldn't really, yeah. Hopefully that helps.
okay. I see what I see, I guess. Yeah.
That's good. Okay. Yes.
Oh.
How are you all doing so far? I think some of you, if not most of you, have already completed your staining, correct? Yes? Who else hasn't completed their staining or who's in the process of completing it? Okay, okay. Okay. So here's another idea for all of you. Some of you have already skipped the step of essentially completing your smear and being able to visualize your organisms. But what I want you to bear in mind is the following. You have sample slides found. Oh. Okay, I'll go here. So here's what you're going to need to see. And I think it'll help having the following slides as demos. So you'll notice some exercises refer to actually visualizing different slides. These are the slides that you have actually for all of you to use as demos. So let's say for whatever reason you can't see anything on your microscope, or you want to see and compare how different microbes look. This is where you can. You have to do whatever you want. So what I would say is have a look, return them back to us and have a look and see if different microorganisms before you leave the end of phase one. So have a look. If you're struggling with things, have a look and see if you're able to compare. And then I'll always have this oil immersion or immersion oil patented after 100x. So after 100x. So just bear that in mind if you want.
It's my thought.
How's everyone doing thus far?
Wait, can you look at my computer?
Not concentrate.
Yeah, I think you're. Okay, sorry.
Thank you so much.
Yeah, just one drop. It's again, good. And then.
Everyone else had operated microscope, right? No? Okay, so I'll go back then. In just a moment, we have 2 minutes.
Mine's good like that. Are you like, what? Yeah.
Who's using a flame right now? Who needs to? Because I'm smelling. If it's left, I can only use it when it's on, or keep it on when you're I'm very much setting it on. In terms of operating a microscope, I apologize, that, it takes a bit of time, and, a bit of, you know, strategy of being able to understand, but what you can also do is use slides to reference at the time. I'll go back to the slide on how you operate, or hopefully if you need to come in directly, I'll show you. And about three, we're following two more questions. So, you can see that there's one. Everyone, if you could please pay attention, I just have a few other important reminders for all of you. Some of you are asking about how you operate your microscope. So just bear in mind that you have a few things that you need to take into account. You have your microscope like the one I showed in the middle here. And when you turn it on, right, you have a few things to work with. The light intensity, right, of your microscope. Some of them, like this one, is flickering, but the point is yours ideally shouldn't flicker. When you turn it on, you have your stage holder here for your specimen, and you can adjust left and right, up and down, for the different, X and Y, the location of the line. The torch adjustment knob adjusts the top to the bottom, rapidly as it said, my adjustment knob does it more sharply. But for many of you, I think it's really just being able to see and operate it more carefully. So what I mean is like,
It should be like a procedure.
and who else hasn't seen anything wrong with smears? Just for clearing up any outstanding items, which are And I'll get out to everyone else shortly. I just want to make sure you're able to start.
No worries, sorry. It should be more
That's right. All three doctors. Okay. Actually, here's the good. So how we are here now, you have to take a mixture or like you have to actually show yeah, so the you see only on those.
Yeah.
So, yeah, you need it. Oh, I ordered. This was I'm sure you can show it, but there's one. Apart from the people I've spoken to recently, how many of you are done or need more help? Or actually need help in this time? Okay, 100%. Don't worry, I'll help. And then this time, okay. So, okay, I'll go to all of you. I'll go to all of you. I'll go to you. There's a
Oh, no, that's what it is. That's fine. Should be the same.
Actually, it depends on sometimes the different materials. It can be designed. At times, they actually providing more structure like that. So zoom in and see if you're able to identify origin cluster, rather than like a big bulky guy. Okay.
It looks like it.
So just one last clarification. Everyone inside of the see up and left.
Yeah. Okay, it's fine. Yeah, like the.
Just one breath. And then
Also, just to double check, as long as it's in the oh,
Oh my God.
Great. Okay, I'll go back to letting everyone else know here on the thing. You have how they're arranged essentially. So like in pairs and I don't want to say solo, but individually, pairs. There's clusters. There's multiple as well. Like I'll show you in some time. Chains can work, and the reason I didn't say spirals is because individual spirals would more likely look like this, individual rods, right? Which is not really the case. So what yours is more like that's how you can describe it. Yeah. So everyone, I just want to say that I'm very grateful that we are actually pretty much completing two main components, critical components of today's lab, and one more mainly to go with the ocular micrometer. We also are going to try to do the hanging drops shortly, but I want you to be able to, in the next 20 minutes, complete the ocular micrometer in groups. So after you've drawn pretty much your bacterium or visualized it, then here are the few things that you need to take into account. As asked earlier, you need to be able to describe the morphology in like a figure caption of your bacterium, describing the magnification, the type of bacteria that you used, whether it was in solid or liquid agar ideally. And then I should write them. Actually, just keep them in mind. Magnification, the morphology, the size, which we're going to talk about shortly, and the color as well. And then that way you'll be able to be as descriptive as possible for your figure caption. Has everyone been able to finally see their specimen, at least one of them? And then hopefully if you can, we can explore the other slides another time. But now we really want to take heed on the ocular micrometer. So if you haven't done so, take a photo or just draw out the bacterium that you have, and then have a listen to what I'm about to say. So, oh, and it's already written down there, which is nice. But let's have a look here on the slide itself. You have your eyepiece, right? And I apologize. Normally, I don't do this setup, but hopefully you'll get an idea as to how you can efficiently or hopefully better appreciate and complete the rest of today's practical. In that, now that you understand a bit more into what a microscope entails, like how you adjust certain pieces of your microscope, and how you're able to also visualize different specimens from your own and the slides included. The next step is actually measuring their sizes, right? Because you want to be able to compare different bacterial specimens and potentially down the road, compare them with other novel bacterium or viruses or other microbes that could be of clinical importance globally. So think about that for a bit, digest that, and then look at this discussion question. So imagine you have your eyepiece micrometer. If you look normally on your left hand or your left eyepiece, you'll notice that there's a ruler that sticks out, right? And that's your eyepiece micrometer, i.e. it gives you a given range as to how large relatively your microbes are. Now, to use what you see here, a stage micrometer in groups, i.e. separated in order. You're going to be using separated for as well. You're going to each take use of one of these ocular micrometers. They are, so you've done the more difficult task of looking at bacteria. This time you're just looking at a ruler, and that's your ocular micrometer or stage micrometer. The point is that when you just place it into your microscope, you really just want to align the ruler that you see here which is 0 that you see on your eyepiece. And when you do that, you'll be able to, and actually, let's pause there. You guys can grab them there as a group. That's all you have. Okay, you guys can change your seats. I think has already gone one. We have it. And you guys can surround each other as a group, or you guys can pay attention and then do other parts as a period. So, so just bear in mind that don't throw it away. And then what I want you to do now is really just use the microscope, place it into any of the microscopes that you have handy with you, and then essentially try to find a way to align both rulers at the 0 mark. Any questions on that? Is that explanatory? Any problems, any concerns? Forgive me because for context, I TA for labs, so it's a bit overwhelming for me personally. So I hope I don't come off in a way that you're left clueless, essentially. So let me know at any time. I want to make sure you're able to complete this lab with success. So don't worry. So you guys can make your way to using the ocular micrometer now. Have one microscope that's left without your specimens if you're done, or you can use your own if you're done with your own, and then you can align them. So you can place it either on your feet or essentially your goal is to align the ruler and align it with your eyepiece. How do you feel, Toast? Just pass your This one actually isn't good. I would recommend. Work. If you also, if you're having trouble fixing your microscope for whatever reason, let me know. I can help either ask the lab technician to repair yours or grant you a new one. So just let me know. Do you have a question? One.
Yeah. I try using it. Yeah.
I definitely see something. I'm not sure. But you can help me understand.
How are you guys? Okay, good. Any other questions?
Yes, we can see anything. Now we
Okay, that's. Right. But you got to see your study, your brain back here.
I saw, I don't know why I can see it.
Sometimes it's very small.
I used 40.
Yeah. Start with the pen. So has everyone aligned as a group their rulers? No? Not yet? Is it a bit difficult to do so, or like are you able to find a ruler or no? What are the problems that you guys are seeing? You have problems? You good? Yeah? Group in the middle, left side. Are you guys having any problems?
Yeah, we could look for it.
And it's a little bit of people.
If you guys look for it. But it's good. They are
I just started with
Okay, start.
those are the bacteria, those are the ones.
Okay, should I do the micrometers first and after?
Yeah.
Okay, so just make it safer now.
That's what I would say, shut off and then you can turn it on.
So how do you touch it?
We need to draw.
Just take pictures and like that, it's right? So, you guys want to see it? Not that. Not just one. Where's your most stuff?
Yeah, we'll do that first one. How many? Yeah, there was. We just have to address like the, you know. Oh, I see that. Okay, just a minute.
Okay. How's everyone doing so far? Everyone able to align their micrometers? No?
Yes.
Okay, you guys have done it? You guys have done it? Is that a hand raised or like a good? Okay, I'll check you. Okay, okay, so I'll go to the two of you. So these two groups.
Oh, yeah.
Right? Yeah. On the,
You're here.
You may just want to find it just right now. It should be.
yeah, wherever it finds your class.
Just bear in mind the magnification you're using though. They may be different depending on the groups you're using.
Do we need to draw?
The bacteria, yes, but not the ocular micrometer. Yeah, yeah. Yeah, yeah. It's so I will explain. It's so what we're doing now, I'm trying to break it down to make it simpler for you. Once we get this down, overlapping the micrometers, the counting and calculation of your sizes will be a breeze. Okay, so don't worry about it. But really, I just want to make sure that you're able to see and visualize this from your microscope.
On the right side.
So we have two options. You can either add the different else. Actually, I like to help you. Because if you keep it as it is, that's 4x. We're going to talk. I'll show you what. I just want to make sure everything's overlap.
Yeah.
Able to see what we need to do now.
This ours, like, I think it's like how little it disrupts it.
If you, I think
If it's like 5 micrometer here, about paper.
it's not my concept.
Lessons.
Yeah, the one that you, the one. So don't worry. Now you have your micrometers overlapping. What's next? So have a look at the slide shown here. Let's say you're looking at your OUs, which is your stage micrometer, aka your ocular micrometer, right? So just bear that in mind. The stage one is your ocular one. And so you have known digits on top from the stage, right? From your eyepiece, right? Or sorry, yes, from your eyepiece. And let's say you count up to 20, or wherever these two rulers also intersect beyond just 0, you want to look to where the given values are. So have, I would say, at least one member of your group looking at the ocular micrometer right now, seeing where the two rulers also intersect, not just at 0, but at another location. For example, the eyepiece here may intersect at 20, and at from the eyepiece, but for the ocular stage micrometer, if you count, it'll be 5, 10, and then 18. Sorry, 5, 10, 13.
So 13 OUs or OCUs, for simplicity, we'll say 13 OUs intersects with 20 shown here in the eyepiece. Now, 20 is your hallmark, right? And I want to maybe sorry, let me clarify. Sorry, 20 OU is here, and then 13 divisions, stage divisions, is shown here in intersecting, right, from the diagram. Now, the value of what this 13 represents is 13 times 10 microns. Each small division equals 10 microns or 10 micrometers, which is where this unit comes in. 0.01 millimeters equals 10 micrometers. And so when you reach 13 here, that's 13 times 10 because each of them represents 10. So that's 130 micrometers. So if we do the math, 30 micrometers equals 20 OU, 20 divisions on your eyepiece, right? Your ocular micrometer. Sorry. The words are very similar in like hindsight, but the point is that the 20 represents your OU. And so now if you wanted to find out how much each OU is, to answer your question like, oh, these microbes are so small, how are you going to find out essentially how much they, how long they weigh, or sorry, how long they are essentially? The idea is that you have OU as a reference. Your OU, once again, is the eyepiece micrometer, right? Your ocular micrometer. And so because you know 20 OU equals 130 or 13 stage divisions, you simply do the math of dividing 130 by 20. And that's going to give you 6.5 micrometers per OU. That's your calibration factor for whatever magnification you are. What I mean by that is that let's say you're at 4 times mag, right? So if let's say you're at 4 times mag here, and you wanted to determine, actually, I won't even continue further. Just imagine what I would say is continue up to the step that I just mentioned, and then I'll talk about how you're going to calculate your bacterium after. Just really understand this concept. Up to here, of how you're going to see where those rulers intersect or overlap, right? Let's say it's 30 and here at 20, or 60 and 40, etc., right? Then when you find out where they overlap, simply divide how many OUs you have by the total amount of microns, right? By the stage division, and then you'll be able to find out your calibration factor. And that's how you calibrate your ocular micrometer. Any questions on that? No. Was that a bit difficult to understand? No? Okay. Just one. Just one. That's right. So you're gonna, once you're done this step, you just use the same magnification that your group is on to look back at your own slides and look at any like microbe really. And you could just guesstimate, okay, let's say it's 0.2 microns. 0.2 OU. Then simply you're dividing this by 100, right? Because 0.2 is 100 times less than 20. So then your answer for how long or how big your bacteria is essentially 100 times less than this. So just bear in mind, what you really need to do for now is the first step of finding out where next do the two rulers overlap and calculate based on that ratio of how much one OU is at your given magnification. Afterwards, we'll talk about how you calculate your sizes. Pretty much what I said, but focus on that first step for now, finding out where they overlap. Can everyone do that for me in the next 3 minutes? Yep.
No, not 2:30. 45.
Yeah.
If you could do more magnifications, because it's very simple for you now, to go from 10 to 40, that would be amazing. If you don't have time, just let me know. But in the meantime, at least find it for one mag and let me know. Raise your hand or just tell me, say like, Muhammad, I found that it's like 5.5 microns per OU at 40 mag, right? 40 mag. You say something like that. In about a few minutes, I'll ask everyone from each group to just let me know. That's good. I'll just see this. So you can continue.
It's a little more.
So I just wanted to get any outcome box given that I didn't go back to your review. Anything else that you explain?
So the joint from the pure. Sorry.
Anything else?
Can't find the and maybe search for. Yep.
Yeah.
You all have that. I'll show you after. I'll show you after, and if you can. It's difficult to have it.
Okay.
Huh?
I think it's so far.
My advice is,
Let's go back. You do not.
we're not doing it.
Yeah.
So how's everyone doing thus far? Okay.
And see if there's. Yes.
Oh, yeah.
Yes.
No worries.
Yeah.
Sorry.
Or If you say for yeah. I think. What's the, that's
So now everyone has pretty much done their maths for the optical micrometer. That's okay. That's okay. Because the next step is, and everyone, I just want to make sure you're paying attention. Everyone, hello? So let's make sure we're paying attention. We're pretty much done almost the practical, right? So well done, you guys. I just want to make sure though that we finish off well and complete the following. So imagine, as you can see from the figure here, you have two OUs, right? This is now the stage where, okay, once you've calculated the number of stage divisions, which is times 10 for microns divided by the number that you see from your eyepiece. Once you get that done, you move on to your specimen, because now we're moving from theoretical mathematics now to practice in biology. So think about it like this. If your microbe, let's say, is 3 OU, and let's say, and you have to be specific on your magnification, right? Because different mags can have different calibration factors. So that could also be something to bear in mind for your lab exam. If it's at 3 OU, let's say at 10x, right? And I think, so mine was at 4x. Okay, so let's say, actually, let's stay at 4x. Let's say now I found a bacterium, B. subtilis, and I noticed that it was 3 OU. If it's at 3 OU size, then simply I'm multiplying that calibration factor, which is 6.5, to determine the total length or diameter of your bacterium, which in this case is 19.5 microns or micrometers. Both are the same, just used in different ways. So that's how you essentially find out your bacterial size. So I just want everyone in the next five minutes, honestly, very simple. Just go back to your slide, look anywhere. It could be any bacterium that you had at the given mag that you were looking at, and then tell me what your OU are, or write down what your OU is, and then determine then your size of your bacterium. When you're done, raise your hand. So then we can go to the very last exercise for today. Yeah.
On the calibration, so you change, I thought it would do that.
And you only need to do that max true magnification for calibration. So don't worry if you haven't all work. You need to do what we do now. And then
Oh,
so you don't need to go
yeah.
That's like walking in 100. Six point five. And then if that is not, like 6.5.
POU. That's POU, and then multiply by 6.5. That gives you 19. After this, hopefully in the next minute or so, you can be done with measuring your OU, determining the actual size of your bacterium from the OU or ocular units that you have. And then we're going to end off the last 15 minutes of class with the hanging drop, which you could also do in groups like the groups that you've had now. So if your group isn't done, have members of your group focus on the OUs, right? Determine the calibration if they haven't done so. Determine the size of your specimen, right? If you also haven't taken photos of or drawn your bacterium, this is your time to do so. You can also take a photo if you need to, but just bear that in mind that in your the results of your post lab, I need you to be able to show it to me for next week. Your results, your discussion questions from the post lab, in the manual and PowerPoint with the questions that we've touched upon, but also with regards to what you saw and what you would have expected from the bacterium that you investigated. Bear that in mind, as well as with the results. The figure captions need to be as descriptive as possible with the magnification, the bacteria you use, the morphology, the size that we're talking about now, and their arrangement. Yes. For a post lab? That's okay. That's okay, no problem. It may even be a slide here. Actually, it's not. It's not. So it's okay. I'll share them. I'll say them one more time at the end of today's class. That may be better. I'll give you a few more seconds. Don't worry about like drawing it right now. Take a photo. For a bacterium or the calibration? You already did one over.
Yeah.
So it's. So I need us to pay attention because I don't want you to be rushing in the last minute of class. So start splitting up in your groups to focus on the hanging drop. The hanging drop we only really need to do as groups as well. You could do them as pairs if your entire group is done, or you could just do it as pairs and then let the remaining amount of people help focus on their OU calibration for their sizes. Yes.
A question of the microscope.
Okay, I'm gonna first have to go straight to the hanging drop. These are also questions. No. You still need to. But I was saying that people in your group can split into pairs to focus on the hanging drop. But first, we are all.
Yeah.
Okay.
Okay.
All of them have already been.
Yeah.
I wish well, at least of you have already done this, all the I mean, the consolidated, this is like part of the whole class, you don't, I don't think in class, but now.
You want to measure the sides, or measuring just what? How about you talk about the rods, about them together? Just here's one.
This one. If you do not, if you, let's say, two, I'll say, well, let's say you can divide that for the time. So now let's go straight to, sorry, I wanted to mention this as well to answer the question asked before. Your post lab revolves around three main things. Your results from today's lab, the figures that you've taken, or at least taken photos of your microbes, right? Taking an image of them, having a figure caption, making it as detailed as possible. That's part one. Part two is your discussion involving the questions, the answers of everything that I've mentioned thus far, the PowerPoint slides, the lab manual. So bear this in mind, everyone. I just want to make sure you're paying attention. I'm going to repeat this one last time. So let's pay attention. Your post lab revolves around three main parts or themes. Your results from this week, right? The figure caption, the photo that you've taken, or the image that you've described in your lab notebook alongside the caption, making it as detailed as possible, as mentioned. Then you're answering in your discussion, the second part, the questions that you see here, for example, and across the slides of the PowerPoint on Quarkus, also the ones that you see in your lab manual, and then, which is even more important, describing and comparing the results that you have from today and comparing that with what you would have expected. That's pretty much a bulk of what you have for your discussion. It can range from half to one full page in your lab notebook, so just bear that in mind. And then the last part is pretty much your pre-lab for next week. So bear that in mind about like what you need to do, what flowcharts, which need to be as visual as possible, your introduction, and also don't forget, if you haven't shown me your microscope as well, to do so then. That pretty much is your pre-lab. That's pretty much your homework for next week. Any questions on that? No? Okay. And I could stay, well, like technically the lab may prefer me to leave. So what I will say though is, outside of negative staining, the goal was really to focus on simple staining. If you look at the video here, you'll be able to know how to like essentially do the staining, but it's also shown with some templates here in front of me. The hanging drop is the last component that I really want you to focus on. And pretty much as groups until 11:55, or if not earlier, actually, maybe in 10 minutes, because it's much more simpler than you would imagine. I'll show a demo on either side. This week, I'm sure everyone's paying attention. And ask one of you to put on your bus. All of them have that. But in the time to get ready, I'm going to show you guys. So, Everyone who's just now the instruction here. If the people who use the sizes of your don't worry. Just come here, you'll see the hanging drum. So pretty much what you need to do is the following. So let's all pay attention. I'm going to go to that shortly, but I want you to pay attention, everyone. Is that what you have here is your hanging prop. The depressions are above, simply because you won't feel them on this side. The depressions are where you're going to essentially have what looks like that slide here. And what you're formulating is essentially having a cover slip with bacteria on one side, and then gradually on this depression slide. When you're done, you're going to tilt it over such that your gene depression slide is going to be inverted onto where your bacteria are. Don't forget the aseptic technique when you deal with the bacteria. And then it goes back, like once you've inverted it and placed it on your slide with the cover slip, then you just view it on the microscope. Where do you have to look? It's not making any sense. It's just determining if the bacteria move or not. And move in the sense of not undergoing Brownian motion, i.e. they're not, either they're vibrating or they're showing true motility and they're moving in directions like that. So bear that in mind. When you're looking at the light microscope, we're looking only at the edge of your drop. So that way you're able to focus in on that bacterium at the edge of the liquid, which is why you're standing in a drop of water and you're not see your bacteria. But it shouldn't be. See if we could do this in the next 10 minutes. You already have here, and if anyone wants to have a look as well, thank you so much. So everyone on this side, are we have a bunch of comments? Yeah.
This one is the other. This actually is the one that
So anyone else has their particular comments? Anyone starting with the question? Slide, does anyone know how to do it? So you've got one to do. Everyone move into the center. I'm going to be showing you. You guys can continue as is and show the description. I'm just going to show Ameen as well. Just don't forget the depression slide to return back. So here's the depression slide. I'll place it in the middle so you're all seeing information. Which will be good. You can return it back afterwards. Yeah. So, yeah, that's okay. So, okay, so what you do is you pretty much have your smear showing your bacteria. So,
you're in a microphone. Shouldn't you just yell? That you need to put it away. That? Okay. Looking at slides, it's a fire hazard. Okay.
Everyone can have 5 minutes to finish the hanging drop. It shouldn't take too long.
Stop. And I'm practicing alcohol, should never do that. I should want to. Properly the filters. The reason you have to do that is because it doesn't matter. If some folks are, all you do is heat this up. This now becomes a source of liquid.
Everyone just make sure your Bunsen burner is not too high up, right? Make sure that if you want to as well, make your rod vertical. That can also help. Yeah, keep it like at a tilt so that way it's not too close to you, but also such that you're able to maximize the sterilization of your rod. No. You can do it full. Bear in mind when you're dealing with the tube that especially you're not letting it drop into the Bunsen burner. Very important, everyone.
Which kind of part is the it's supposed to go to right.
Start cleaning up, so everybody. We're gonna drink something, do a real quick time, please. Time to clean up.
So actually, last time, okay. As Dr. Brunt said, everyone, it's time actually for us to clean up. So just bear in mind the dump temperature needs to be shut off, and you can begin cleaning up your bench with any unused items. When it comes to the items that you saw on your bench, the white trays, return them back to their former places. Make sure that you clean them up with the slides. The ones that are depression slides, you bring back to the front. Your own slides, you have two options. I think for those that don't want to keep them, they just throw in the Westernite. But the ones that are with ones that you want to keep, for example, just let me know, and you can leave them in the middle. The other important point is for waste, that is like paper, business paper, you can throw them into the garbage or the out place if it's contaminated with coffee. And then we'll end up very shortly. You guys are in the best shape, though. So has everyone been able to look at their bacterial? Yes, right. Don't be forced if you haven't said it.
You just saw the.
Sorry. Oh, solid.
So,
Yeah.
okay, that's great. Everyone hopefully saw
Browninian or true motility in their hanging drop? Yes or no? Who hasn't seen anything, perhaps? If you haven't, just let me know, because I want to make sure you're all able to understand and know and be able to complete the rest of today's practical and your post-lab without being penalized. Browninian motion, once again, is more vibratory, right? They're not moving in a main direction, whereas true motility, you could see a vortex, or sorry, a vector of direction for these bacterium. They're moving in one way, maybe to another, or just in multiple directions overall. But that may be Browninian if it's vibratory. Flagellar stain, you don't have to do it for this week. However, use the questions shown here to be able to discriminate the different types of flagellar arrangement from panels A to E. That pretty much concludes the lab. Motility is next week, so don't worry about that. But if you can complete the rest of the questions that you see here, that would be amazing.
So put this here.
If you want to keep them, yes. Actually, because we don't need them for this week, we can dispose of them. So we'll be able to fill. That's everyone else, make sure that you have the person employed. Yeah. Everyone, hello, everyone. All of you paying attention. We're almost done, and you hopefully will be able to complete the last within a minute. It's very simple. Wipe off your bench, 70% ethanol. If you have extra liquid in your slide tree, dump them into the wash canal. Those are the yellow, whitish scale features. Yeah. And then make sure you bring back any used items, putting the micrometers in their boxes, and as well as in the question slide. Please bring them all back and your own slides. Present slide, do 70% ethanol, wipe again. No problem. And if you have any slides that you want to hold up, you can throw that into the wash canal as well. You guys really want to be able to see what you have, and you have already taken photos of drawing out. Almost. Wash your hands, and before you please let me know, because I just want to make sure everyone's ready for the full time. And you guys know. I smell something on. Hopefully, no bus spirit is on, right? No one has a bus spirit on, right? Okay. So don't forget ocular micrometers to bring them back. And post lab. Remember the three things I said. I'll send an announcement, yes, but you should think about this right now. What are the implications of my results? what are the questions that I have to answer, and what's next for me for week 2. Start thinking like that, start thinking in advance, and hopefully you'll be able to excel throughout the many practicals we have left with each other. Thank you so much, everyone. Have a great rest of your day, and looking forward to seeing you next week.
Okay. Thank you.
Bye, take care. Don't forget to wash your hands if you can. Yeah, right there. Don't forget to pick your caption for the results. You are ready to go. Long enough. Take care, everyone. So, groups, the one on top, building. Practical, we can wash it, you know, and then you can put some glass in the person. Everyone done? Take care, everyone. Oh. What I showed earlier, I'll just open it up again. Right there, pretty much like that.
Okay.
I don't want to get in trouble. The water bottles are. You're gonna have to keep it outside. Anyone else having any questions, any concerns? Has everyone placed the article in the comments back? Did the questions slide?
Yeah. No problem. I really appreciate your time. Any questions? Perfect. So, the microscope or your, no, sorry, your, okay.
Like not really microscope.
Like, you don't have to draw it like that, but definitely want to do it.
Okay, I can make you a picture of.
You can do it with the next.
Oh, yeah.
But try to complete all of this as much as you can.
Okay.
Perfect. Oh, yeah, no problem. Yeah.
So, this is a random question, like, you know, how many things that you can figure out challenge, how do you recommend at the time of the
That's a very important question. Great question. First off, washing our hands first. And then afterwards you'll be able to use the phone. If you already use your phone, there's two options. You could sanitize your phone with a chem wipe. And sometimes like you want to do it a modest amount because if you do it too much, it may actually affect your screen as well. But try to do it like I would say a modest amount is okay.
use some academic camera, or just in like,
Sorry, a chem wipe with ethanol. Like the ethanol that you have, let's say from the sanitizers, like the hand sanitizers that are outside. You could also use them to clean off your phone.
okay, so you just take my phone a lot and.
I know.
Your bacteria.
Yeah, yeah. It's great. It's a great question. Take care, Zara.
Thank you so much. How-
You too. Take care.
Thank you.
Have a great day. Yeah, I don't know if you see the environment. No problem. Have a great rest of the day.
السلام عليكم ورحمة الله وبركاته. بسم الله والحمد لله والصلاة والسلام على رسول الله صلى الله عليه وآله وصحبه وسلم ومن والاه. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. الله تعالى في القرآن الكريم تعالى أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. جزاكم الله خيرا كثيرا. استغفر الله. سبحان الله الحمد لله الذي خلق على نفسي وزنة عرشي ومداد كلماتي. سبحان الله الحمد لله. Okay. Myself, how are you, أخي? Alhamdulillah, man. Great to see you, أخي. Alhamdulillah. SubhanAllah, I'm doing a meeting. And inshallah, I look forward to seeing you for Salat, inshallah. Inshallah. Take care, my dear. Inshallah. I'm going to be praying soon, inshallah. Inshallah. Wa alaikum assalam. SubhanAllah.